Organizer for the Global Society for Genetics and Genome Biology for this academic year. On behalf of GSGGB, I welcome you all to our second part of our two-part series on next-generation sequencing and bioinformatics. Today's presenter is Dr. Huayan Ho. Dr. Ho works in the Wilson Lab at the Hospital for Sick Children, where she has also completed her PhD and does research in medical genetics and bioinformatics. Today, she'll be applying some concepts about next-generation sequencing data that you've learned in the first part of this workshop and using it in the R environment. As stated earlier, please make sure you have R and R Studio downloaded and installed on your computers, as well as the GitHub repository, which we've linked in the chat, as well as the packages at the bottom of the GitHub page. If you have any questions about doing that, please message in the chat box, or send either me or Jed a private message on Zoom. And if you have any questions during the workshop, please feel free to write them in the chat box as well. And we'll also have a brief Q&A session at the end if you have any more questions. And with that, I'll let Dr. Ho take it away.
Right, thanks for the introduction, and thanks for the organizers of GSGGB for inviting me to give this tutorial today. Now, again, if you haven't downloaded, the scripts are available on this GitHub link. I will, hopefully everyone had a chance to download it. You can also find a link in the chat box. Now for today's tutorial, I would spend some time introduce you to next generation sequencing data analysis pipelines. And particularly, our focus is using the R environment for NGS data analysis. So hopefully I'll be able to cover some NGS analysis basics in R. And this would mostly cover the common bioconductor R objects and how to work with them. This would include biostrings, genomic annotation resources, genomic ranges. And then finally, I would give a minimum example of an example NGS workflow that you can conduct completely in R. This is done using example ChIP-seq data. And if you're not familiar with ChIP-seq, it's a method for identifying DNA binding sites across the whole genome for a particular DNA binding protein. We will be able to examine raw sequencing FASTQ files, do a small alignment, peak calling, and visualization of the data, which will cover the sort of the most common NGS workflow steps. So before I get into any the content of the tutorial, if hopefully you are familiar with R, as I'm not going to cover the basics of R or R Studio in this tutorial. And also, if ideally you would have attended the tutorial on Tuesday, where my colleague Michael had introduced different NGS data file types and some of the online resources where you could find NGS data. So I also won't be cover a lot of those basics in this tutorial. However, I will quickly go through what a general or most common next generation sequencing data analysis workflow looks like. You would have done your experiments and you send your libraries to sequencing. After sequencing, usually the sequencing facility people would do demultiplexing, which is assigning reads to their corresponding samples. And this is basically the step where you would have received your data. They would become in this file format called FASTQ files. And this is the so-called raw sequencing data format. From there, the first step what we want to do is some kind of quality control to make sure that our sequencing experiment had worked as expected, that we've got high quality sequencing from the sequencer. In addition, these QC might review certain problems that we might want to clean up the data a little bit. For example, one common problem is that we might be sequencing into the sequencing adapters, and this is the part of the sequence that doesn't come from our genomes, and we might affect our downstream analysis. So oftentimes we would perform adapter trimming to get rid of these sequences. In addition, we might want to trim off the bad quality bases to improve our alignment accuracy. Now, once we have this processed FASTQ file, one very common step is to do alignment, which means aligning these sequencing reads to an known genome so that we know from where in the genome these reads come from. This results in the file types commonly referred to as either SIME file or its binary version, BIME file. And alternatively, what we can do is we can assemble these processed reads into either the whole genome or the transcriptome when we don't have a reference genome or transcriptome. Now, when we have these aligned reads, common thing we could do from if we've performed whole genome sequencing or whole exome sequencing is variance calling, and this will result in file format called VCF file, which records genetic variants. If we had performed RNA-seq, for example, a common step might be gene quantification, so we know how many reads map to each gene. If we did a ChIP-seq analysis, which is the profiling of DNA binding protein, then a common step we would do is called peak calling, which is identifying where in the genome these reads cluster. So these would be the regions where the protein most likely bind in the genome. And no matter what kind of analysis we've done or what kind of experiments we've done, a very common thing would be visualization and annotation. So for example, if we've done a ChIP-seq experiment, we want to know what are these protein binding sites? Are they nearby certain genes? Are they overlapping enhancers, promoters? Now these would all be the annotation process. Versus a lot of times when we want to make figures, those genome browser screenshots that you might have seen in the previous session of the tutorial to help people understand our data better. So this is a very, very brief overview of the most common data analysis workflow. And for today's tutorial, we won't be able to cover all of them. However, these hopefully that I would be able to provide you with like a foundation where you can start to explore how to do these different certain types of analysis in R and in other platforms. Now, a question that you might run into when you start your first data analysis is, how am I going to do it? Can I do it on my laptop? Do I have to set up like a cloud computing account? Or can I, if you're in a research facility, they might produce, they might have high performance computing cluster system, which you can use. So the questions might come, do I have to learn coding? Or, do I learn R, Python, whatever? Or can I use some online program to do it? So there are a lot of options for your data analysis. Now, this kind of depends on what is the computational requirements of these different processes. So out of all the processes that we mentioned in the previous slides, some of the usually computational heavy processes would be de novo genome assembly, for example. Alignment, sometimes certain aligners would require more memory than a laptop could handle. Variant calling is another process that usually requires quite a bit of memory. Now these might be the type of processes that might be difficult to do on your laptop as it requires more memory, and the process might take a long time if you don't have multiple threads available. So the most common computational platform, I would say, for NGS analysis would be the command line or the shell, the bash, or whatever you call it, or whatever language you use. So a lot of the tools are available through the command line. I am just going to list a bunch of tools here for each different step, and you can see that you have a lot of options for each of those steps that I've just mentioned. And these are just the most commonly used tools that I could think of. Now, for each step, the red text is the tool that is available as an R package. So in a way, using R, you are able to process through from the beginning to the end through most of these NGS data analysis processes steps. However, for a lot of particular processes, the options available in R may or may not be the best or the most commonly used approach. Now, for example, for alignment, if you have short reads, the most frequently used aligners are BWA or bowtie. You can do it in R, but these packages are relatively less used. And most of the times you want to go with, the most frequently used tools in the field. So there's sort of a trade-off there that, whether you want to stay in R for all your processes, or you would spend a little bit more time learning and try to get the best tool available out there. However, after you've processed these sequencing reads, essentially after alignment or after variant calling, then the downstream analysis, a lot of the downstream analysis are totally available in R, and this includes some of the most commonly used packages, for example, for RNA-seq, identifying differentially expressed genes. DE-seq and EdgeR lemma are among the most commonly used tools, and they're all available as R packages. So again, using R and being able to know all the basics for NGS analysis in R is still very useful, no matter what tools you choose for these pre-processing steps. So we've got a question about Galaxy, so I'm gonna get to Galaxy in 1 second. Now, how do you implement a bioinformatics pipeline? Let's say I went through the literature, or I followed someone's suggestion. I picked out the tool I want to use for each of these steps. How do I actually put them together into a pipeline? Now, what are pipelines? They're basically just a workflow that chains different tools together. And the advantage of a pipeline is you would be able to perform your analysis in a reproducible, efficient, and scalable way. Imagine you're doing everything manually. If you have one file, that doesn't seem like a problem. But if you have 100 files, which is very common in today's NGS world, you might want to have a strategy where you can just click a button and not having to repeat the same thing 100 times. Now, there are different methods to construct such a pipeline. If you use these command line tools or use R packages, you could attach these different steps together using essentially any programming language. Any language would do. Certain languages are better than others in terms of writing a pipeline. For example, I personally think Python is way better than R in terms of putting together a pipeline. A more ideal way might be using a so-called workflow management system. Now, these are basically systems, or you could call them languages, that are developed to write pipelines. Again, I'm listing some of the most commonly used ones: workflow description language, or WADL, Nextflow, or SnakeMake, which is Python-based. You can use all your Python functions and codes together with SnakeMake. The advantage of these workflow management systems is that it takes away a lot of the trouble you might have when you manually write a pipeline in programming language. For example, it could handle resources distribution, like you want some part of a pipeline for a certain tool, you want more memory or more threads going to it, more computational resources going to it, or your step 3 depends on step 2. And all these workflow management systems will be able to handle that automatically without you having to write the code yourself. So this is definitely something that you want to look into if you're dedicated to developing bioinformatics pipelines. Now, there are also web-based solutions, and the most popular and I think the best system is Galaxy. So I'm just going to jump out a little bit and actually come to Galaxy's page. The URL is usegalaxy.org. So this is an open source web platform that allows the construction of reproducible, scalable bioinformatics pipelines. And you can see that it provides you, so on the left here, this panel provides all the tools that are available through Galaxy. It has a very comprehensive collection of tools, including everything that I was talking about earlier, organized mostly by different functions. So for example, if you click on RNA-seq, you can see that there are all these different tools that are available for different steps of RNA-seq. For example, RNA-star is the most popular aligner for RNA-seq data. Feature counts for quantification, for gene quantification. I'm not signed in. So once you sign in, you have a history that can be saved. So next time you come back, you can continue to use your history. So sort of come back to the question in the chat. The tools that are available through Galaxy should be basically exactly the same as the standalone tools. So for example, FastQC in Galaxy should be the same as FastQC that you download from somewhere else, like the actual FastQC software. The only different, it might be the different versions that Galaxy, or the different versions that has been implemented for Galaxy versus the software might be, the newest version might not have been implemented for Galaxy. And for some of the more complicated softwares, Galaxies may or may not have all the options you have when you use the original software. However, this provides you a very excellent platform where it is also cloud-based, so you don't have to worry about the computing power of your own laptop. You could do very heavy computing on here. So if you don't have the resource, you don't have a powerful enough laptop, or you don't have enough coding skills to put together your pipeline programmatically, this is a very excellent resource. And also sometimes you just want to do one simple analysis and you don't want to have to go into the trouble of installing the tools. This is also a great resource. Right. So jumping back, I'll say that, whoops. A lot of the times I personally end up using a somewhat hybrid approach for my data analysis, where for reads processing or alignment or all these these steps that requires heavier computing, I would put them together into a pipeline and run it on the high performance computing cluster or on a more powerful computer. And once I get results that are more manageable, I do my downstream analysis or more customized analysis in R. Right, so for today's tutorial, we'll cover some of the very basics for you to do NGS analysis in R. Now I will jump out of my slides and go to my RStudio. If you have R in RStudio installed and have the scripts downloaded, you can all just open up the scripts. So I am just gonna open, I just downloaded the scripts. I downloaded the GitHub repo as a zip file, and you just unzip it. And inside you should be able to find all these different R scripts. So just open the first one. I apologize that the scripts were developed for another tutorial earlier, so you can just ignore the text there. I put on some links to some useful resources and also some from which I took some code to build this tutorial. If you have already installed the packages, you can ignore the package installation step. If you haven't, please run these 2 lines of code and install all these packages that will be used in this tutorial. If you do not manage, if you can't install the packages for some reason, or if you missed certain steps running the scripts, don't worry about it, because everything, like you could just follow my tutorial for now and then run the code yourself later on. I realize it might be difficult to to follow the tutorial, watch the tutorial and run your code at the same time if you don't have two screens. But don't worry about it, that scripts are all here and you're able to read them later as well. So the first thing I want you to do is to set the working directory. Now this doesn't work anymore. Now my scripts were downloaded on desktop. So I'm just gonna go into this directory, and that just makes, from there, the scripts should all work. Like the data that you need for this tutorial should all be in that directory. All right. So for the first part of the tutorial, we are going to talk about strings in base R. Because when you think about NGS analysis or next generation sequencing, it really comes down to two things: it's strings and numbers. So you care about the actual DNA or RNA sequence, the actual nucleotide, which translates to characters in R. And you also care about genomic coordinates, chromosome 1 from this position to that position, which just translates to numbers. So essentially manipulating or analyzing NGS data comes down to being able to analyze characters and manipulate these numeric ranges, coordinates. So that's why these being able to manipulate strings in R is very fundamental when it comes to NGS analysis. Now, to begin, we are just going to create some random DNA sequences. So we just ran these two lines. You can see here what I'm doing is simply sampling the four letters, and attaching them together. And by doing that, I created some sequences. Now, these are just very random sequences that are of lengths between 20 to 30 base pairs. Now, there are a lot of, we'll just assume that these are our reads from our sequencing analysis. Now, there are a lot of functions available in base R for string manipulation. Now, for example, I'm again just listing some of the functions in base R. You don't need any packages to run these. Now you can calculate or count the number of characters in each string. You can use this function called substr, substring, to subset the strings. And I'm getting only the characters that starts at 3 and stops at 5. You could do pattern matching using this function called grab. Now I'm just grabbing, whatever sequence that has the CAC pattern. All three sequences have this CAC. However, this just tells you which sequences contain this particular pattern. It doesn't tell you where in the sequence this pattern was found. And then you have to use another function for that, which is grabbing the regular expression. So this function finds all the matching patterns in all these strings. Well, the result is in a somewhat weird format. It's very hard to read, but you can tell that this is a list. And for each element in the list is the result of this pattern matching or this pattern searching. So for the first sequence, it doesn't have this AA sequence, so everything is -1. And this is the second sequence. So we could find this AA pattern starting from position 10, and the length of the match is 2 base pairs, and it's a character. So it works, but it takes a bit of effort to parse the results. You could also do string substitution, like say I want to change all the AAs to NNs, use this gsub function. So this is our original sequence, and this is sequence after our substitution. You can see that this AA has been changed to NN here. And if you want to learn more about all of these functions, you can do question mark, and you can see that the series of functions are all listed here, and you can read specifically about every one of them. Now, as I said, this works, like this is fine. However, when we deal with biologically meaningful strings, we want to be able to have some help. Like we want to be able to manage them in an easier way. So this is where the package biostring comes in. And in this package, it defines a different class called X string. So again, we can just do question mark X string and then see all about it. Very briefly, it's a general container, it's a general class of objects for storing biologically meaningful strings. And it is able to store DNA string, RNA string, and amino acid string. So all the common sequences that we would run into. Again, you can read all the details. I'm going to skip here. And there's also X string set, which holds multiple strings. So instead of one string, you could put, your whole sequencing library in here as a X string set object. Now, let's see what these DNA strings look like. So we got these random sequences, the 6 we got here. We can simply convert it into DNA string set by using this function. Now let's see what they look like. So this is a new feature I noticed with R 4.0 is that it automatically colors the different nucleides. So it's pretty nice. It looks nice. Not sure if it helps too much, but you can see that now this is a DNA string set object of length 3. And for each sequence, it tells you the length of the sequence and the actual sequence. If your sequence is super long, let's say 1,000 base pairs, it's not going to display 1,000 base pairs. It's only going to show you some of the sequence, so it makes it easier to display on the screen. Again, we have a lot of manipulation methods when it comes to these biostrings. So for example, we can get the length of the strings using width. We could, again, subset these strings. Let's say, again, I only want the characters from position number 3 to position number 5. You could use this function called subseq. And it looks, again, a bit nicer than the previous results, as after the subsetting, you still get a DNA string set. So the type of object is still the same. Now you still see this coloring and everything. It also has a little bit more powerful pattern matching methods. So for example, using this V pattern matching, I'm still looking for the AA sequence in the 3 strings. And the results is, I think, a little bit more readable than the base R pattern matching methods. So for the first sequence, we don't have an AA pattern. For the second sequence, we got an I range object that starts from position 10 and ends at position 11 with a width of 2. So this is a little bit easier to both read and also extract for your subsequent analysis. Now, in addition, it also allows for a little more flexible options. So for example, it can allow for some mismatches. So let's say I am searching the CAA string or CAA pattern, allowing for maximum one mismatch. And I'm assigning the results to this p-match result object. So after running this line, what the p-match result looks like. It's essentially the same as before, but now because of the one mismatch, we got a lot more hits. So you could imagine that you could use this function for finding motifs in your DNA or RNA or amino acid sequences. Now, to be able to see what are the methods available for this particular type of object, you can use this function methods and say class equals X string sets. And it comes up with 166 methods that you could apply on this type of data. Of course, we're not going to go through all of them one by one, but just a quick look through them, you see something that, something that seems quite useful, such as alphabet frequency. That sounds useful. compare strings might be useful. Here I have some examples of the useful functions that makes our life a lot easier. For example, there's a reverse complement function, which simply gives you the reverse complement of the sequences that you have. You could, of course, write a function yourself to get the reverse complements. It's not that difficult, but having such functions handy is one advantage of using packages like that. You can simply calculate letter frequencies. So if you want to know the GC content of these sequences, you could use this function. And you can translate your DNA sequence into amino acid sequences. Now, because I just got random sequences, so there's a lot of warning messages as the numbers don't match. But you could do that if you have the actual DNA sequence. And again, it's very convenient. You could also trim off flanking patterns. So as I was talking about earlier, sometimes you would have sequencing adapters at the end of the reads and you want to get rid of those. So you could, if it's a simple situation, you could actually achieve that in using this package. So I'm going to create some extra patterns by just attaching this AATTA sequence to the end of my sequences. So you can see now that at the end of the sequences, there's an extra AATTA. And then I can use this trim LR pattern function to get rid of it. So you can see that it just trimmed off the extra sequence that I attached. Of course, here you have options to allowing mismatches, allowing indels or deletions. So a little bit flexible. Than just using the base R functions to achieve that. And then finally, what if we create a RNA string instead of DNA string? So you can see that is automatically translates all the T's, converts all the T's to U's. This is because internally this package defines a set of alphabets for the different types of data. So you can use alphabet function to see the alphabets available for either RNA or DNA or amino acid. I don't have an example here, but these are all the available characters for this type of data, RNA string or DNA string. The minus here actually means gap, plus means masked position, and then dot means, I think, missing data or something. So what this helps you is, again, it helps you to predefine the alphabet that you could use for certain type of data. So again, making a lot of operations convenient. So this is all we have for biostrings. It doesn't cover all the functions available in the package, and it also doesn't cover all the things you could do with these biostrings. And you could read that these additional resources that I put in here, or just Google and read more tutorials to learn more about them. Right. So if you have any questions, just feel free to put in the chat. And meanwhile, I'm just going to open up the next section. So in the next section, we are going to talk about genome annotation resources that are available in R or through the R environment. So at the last tutorial, we showed how to access some genomic resources, publicly available genomic resources. So for example, if you have sequencing data from human and you need to do your alignment, then you have to get the human genome and you have to know gene annotations of the certain human genome, where in the genome does the gene locate to, how many transcripts associated with the gene. You might want to have additional annotations, like what are the Go terms that are associated with these genes. So all of these information are available online. And you can get them through many different ways. You could certainly just download each individual resources and then compile sort of like a database yourself. But this process has been made easier through R, as there are a lot of these annotation databases that are already pre-compiled for you and are pretty easily accessible. So I'm going to walk you through some of the most commonly used annotation packages in R. So to start, all of these are available through Bioconductor, which is a repository for NGS for NGS analysis, for packages focusing on NGS analysis. So in Bioconductor, a very common type of object for storing these annotations is called annotation DBI object. And the most commonly seen specific types of objects are ORCDB and TXDB. So I'm going to start with these two types. So ORCDB are a database that stores organism level information and is gene-centric. So it stores the mapping between like gene identifiers and other information, such as like Go terms I was talking about. So if you have installed it, let's load this library. And from the name, you can tell that this is a human ORCDB database. Now there are some core methods for these type of annotation DB objects to access the information that's stored in this database. Now, when we talk about database, you could think of it as a giant table or multiple giant tables where the columns are linked in certain ways. So in this case, is essentially, you could think of it as essentially one table. So by using the function columns, you could see the columns of this table. So these are the information that are stored in this database. Some of them are pretty intuitive, like ensemble ID, entries ID, Go terms, gene symbol, and some other gene ID systems and gene annotation resources. Now, when you query a database, oftentimes you use these keys, and you could use the key type function to see what are the types of keys that are available for you to query this database. Now, if we do key type org HS DB, you see that it's essentially the same as the column. So you could use all of these columns to query this database. You can use the keys function to see the actual value that's stored in this object or in this database. I'm going to only show like the top 6 head keys. And if you just do keys, the default is extracting the entries ID, so they're just numbers. You could specify what key type you want. So for example, I want to see gene symbols instead. So you can say key type equals gene symbol, symbol. And now you get the first several gene symbols that are stored in this ID. Now, how do you actually retrieve information? A core method for all these annotation DB objects is the select method. A lot of packages would have a function called select, particularly because we only have certain packages loaded. When we call select, we should be calling it from the annotation DB packages. Now for select, you would have to provide it with the database you want to query and the keys you want to query. So I want to know everything about TP53. So my keys is TP53 and key type. So this specifies what type of key you're using. Now this is a gene symbol, so my key type is symbol. And obviously you would be choosing from the available key types for that database. And then finally, the columns tells the package or tells the database that what information do you want to retrieve from this query. So if we run this line and take a look at this result, you can see that we got our symbol, which is TP53, our ensemble ID, this ID here, and also information that these are the Go terms ID that are associated with this particular gene. And there is actually quite a few of them. Now, what if I only want to get ensemble IDs? Like I only want my gene symbol and ensemble IDs. There's an alternative method that is called map ID. Now, it's essentially have the same parameters or same arguments as the select method. The only difference is that it'll return your results as a vector. So you can see that now this is a named vector where the names are the ensemble IDs and the actual values are the symbols. So sometimes this is more convenient for certain types of analysis. So this is just an alternative way of retrieving the information and resulting in a vector instead of a data frame. And it only provides a one-to-one mapping, so mapping between your keys and one columns. Okay, so this is the ORG-DB. And if you go back to the keys, you can see that it contains all the information about genes. However, what is missing is it doesn't have the transcripts that are associated with the gene. And it also doesn't tell you where in the genome does it map to. So it doesn't have the coordinates of the gene. Now to get that, we have to use the TXDB objects. So TXDB objects are transcript oriented, and its main purpose is to map between genes and transcripts and their genomic intervals. So I am loading this particular library, and it's very clear by the name that this is, again, human data, and it's getting known genes from UCSC, and the genome version is HD19. There are other packages for different gene annotation resources or different genome assembly that you can download from Bioconductor. And I'm just going to assign it to an object called TXDB because it's easier to type. So again, you could use columns, TXDB, to see what are the columns available in this particular database. Now we have chromosome and start, strand, all these additional information about the particular position of different transcripts. So now instead of gene IDs, now we have transcript IDs, transcript names, and everything is transcript based. You could, because this is also a annotation DBI object, so you can still use this select method on it. So now let's first get some random genes. We've got some genes. These are genes. Now this is again the entries ID, which is as numbers. So it's not very informative. Again, we use the select method, and we want to know what transcripts are associated with these genes and what are the locations of these transcripts. So by doing that, we get that for these particular genes, these are the transcript names, transcript locations, chromosome, start, stop, and strand. So in addition to the select method, the universal select method, there are some TXDB specific methods that uses the genomic features package. So this is another resource that you could look up. You can just find this on Bioconductor and read the vignettes or read the menu documentation of the package to learn about all its different functions. But it provides some very convenient functions that extracts data from the TXDB objects. Now, for example, they can get all the transcripts from TXDB. So this is going to take a little bit of time. Yeah, so we got the top 10 transcript from TXDB. You can see that this is in a G range object. I'll get into the specific object a little bit later. but it's quite intuitive. You get the sequencing names, the coordinates, the strand, and the transcript ID, and a transcript name. The transcript name, again, it's not a very informative ID. Like just by looking at it, you won't match it with the gene symbols, which we're more familiar with. I believe this is the UCSC annotation. But in anyway, we got the information. Some other very useful methods, for example, exons by. So you can get all the exons that are annotated for a certain gene. This is going to take a little bit of time, but you can see that I specified by gene, which means extract the exons by gene. Now this is a G-range list object. And again, this is gene with UNTRUS ID 1. You can see that these are all the exons that are annotated for the gene. And it shows you the chromosome start and stop for this particular exon and ID and strand. So if you want to, study the specific exons of all the genes, you could simply use this function. So there's a question, when you say top 10 genes, is the top 10? Yeah, so when I say top 10, it's just the top 10 that are stored in this database. So like, for example, when I do top 2 here, it just got me the genes with ID 1 and ID 10. That's just how it's just the order that is stored in the database. It's not organized by any particular information, such as transcript abundance. So I'm just using this as an example. In real life, you probably won't do something like that. All right, so another very useful function is called promoters. So this function lets you get promoters of your genes or your transcripts. And you can specify, how many base pairs up or downstream you want. You could apply additional filters. There are specific ways to use these filters, could be confusing, but I'm just showing an example here. I only want promoters on chromosome 19. And again, I'm just showing like the top 10. So here, in this case, the top 10 are ordered by genomic coordinates, I believe. So you can see that they're all like relatively smaller number in the genome. But you get, the transcript name and transcript ID, and this would be the up and downstream 3000 base pair from the transcript start site. This is quite useful as if you are involved in doing any kind of genomic analysis. A lot of times you have to define promoters of the gene, for example, or you want to extract, the first 2000 base pair of a gene, or the last exon of a gene, or the 10 kb downstream of gene, which you could all sort of very conveniently do with these built-in functions. There's some even more useful functions, such as 5' UTRs by transcript. So it gets you, so instead of exons, you get you all the 5' UTRs that are annotated in your database, and you can get 3' UTRs or introns. So basically, the genomic features package provides certain useful functions for you to extract useful information or meaningful information from the TXDB object quickly. Now, we could actually, do a little bit of more sophisticated things. That if we are given some genes, we want to find their promoter regions. However, these genes are given to us in the gene symbol format. So again, I'm just getting the top 10 genes that are stored in the database. These are the genes that we have to work with, and we have to find the promoter regions of these genes. Now, it seems tricky because we have the promoter regions stored in this TXDB object, which doesn't have gene symbol information. If we go back to TXDB, we get, we get a gene ID, which is UNTRUS ID, but we don't have gene symbol. So what do? So we have to get the UNTRUS IDs first by using the ORCDB object. So we just say, you know, key types equals symbol and columns equals unctuous ID. So we get back the unctuous IDs. And then we can use the promoter function to find the promoters. And so by running this line, we get the promoters. And we used a filter here and say that we only want the gene IDs that are equals to the untruth IDs that we found for our target genes. So that way, our result would contain this gene ID column because we can specify what columns we want, and we want transcript names and gene IDs. And then finally, we could match the gene symbols back and then add it back to our gene promoters. So I'm running this code very fast because they're just a sort of a quick way to do it. But the end result, you can see that you get the corresponding gene symbols attached to the gene IDs, and now you also have the promoter regions for these genes. Now, this is a really complex process. Like, we want to be able to do this quickly instead of having to map between these two different packages. Now, one solution is actually that you could use these ensemble DB packages, which interestingly, it contains more information. So it contains the gene locations, and it also has all these gene symbols, ensemble IDs, and gene names. So you can very easily map between these different information. So this is actually what I would recommend using if you just want to do something quickly with these known gene annotations. Again, it works basically the same way as these two packages that we talked about. And you can find more about it on the Ensemble DB documentations. And another thing to note is that you could actually make your own TXDB object. So now we are loaded the one with UCSC, that like this gene annotation is put together, curated by UCSC. But if you have a customized gene annotation, you could also make it into TXDB object so that you can manipulate it easier. And there's, again, you can follow this link to see how you can make a customized TXDB object and utilize it. Now, there's another way of incorporating the information from the two packages. Is this called organism DPLYR package, which essentially helps you to pull information from these two different annotation resources or different databases and then merge them together and build another database for you, which has all the information. It sounds complicated. The use of it is relatively intuitive. It's pretty much the same thing. The only difference is you have to initiate a SRC organism object. So this is how you would initiate it. I'm using this TXDB again. And I think it automatically matches the organisms that this TXDB corresponds to. But of course, if you're working with a very rare genome, you may not have these organisms supported, and you may not be able to use this package. We can see the columns of this SRC object that we just created. Now you can see it contains like all the information we have from the two databases that we talked about. We have exon positions, transcript positions. We've got goal terms, all ontologies, gene names, everything. So now you can query this database the same way, but simply just get every information you want. Drawback is this package or this database is a lot slower to query. So you can see that when we run the previous code, it basically executed in seconds, and this one is taking a while. And hopefully it doesn't stuck. Okay, I'm gonna skip explaining this, but you could use the methods from the TXDB objects on this new package or this new database object, and you have stronger ability to create different filters. So I'm gonna skip like the way to create filters. Again, you could read the vignette to see the specific methods. And I'm just gonna jump to this example here where I want all the promoter sequences on chromosome 17. And the filters I am using is TX chromosome equals CHR 17. And by running that, I should be able to get all the promoter sequences, all the promoter regions on chromosome 17. And indeed, we got 300 up, 3000 up, and 3000 downstream from TSS. We got a TX transcript IDs, transcript names, and gene symbols. We're going to use this a little bit later on. So if you could just run this line. Again, I'm going to skip the third method here. It's a little bit more complicated, but it integrates with the DPLYR or the tidyverse system. So you could use some of these tidyverse functions to manipulate your data and organize it in certain ways. And then the third or the fourth annotation package that I want to introduce are called it's called BS genomes. So the previous packages, or the previous annotations, we got our gene annotations, we got our gene coordinates, but we don't have the actual genomic sequence, which is stored in these packages, the BS genome packages. And you can see that these are all the available genome sequences that you can obtain through Bioconductor. Now I have already installed the UCSC HD19 human genome. So you can use this function to see what genome have you installed. If you haven't installed it, you can install it using this command. Otherwise, if you have already installed it, you just load it. And it's got a short form. If you just type H-sapiens, you should get an overview of this package. So this is human genome, provider, UCSC, release date. You have 298 sequences, so this includes some context and in addition to the major chromosomes. You could get an idea of what's stored there by, for example, using seq names to get all the chromosome names or the contact names. These are just like random or uncharacterized fragments. You could access the sequence of one particular chromosome. For example, this gets you the sequence of chromosome 17, and we can see what it looks like. So again, this is a DNA strain object that is way bigger than anything that we've played with later on. And you can see it's only showing you some letters from the start and end of this very giant string. Now, what I want to do is, I'm using this to obtain information. So I want to actually store this chromosome 17 sequences into a FASTA file, which is a format that we use to store DNA sequences or RNA or amino acid sequences. So to do that in R, we'll go back to the biostring packages, and we actually have to convert it into a DNA string set object, even though there's only one string, but that's just how the package or the method requires. And we'll give it a name. We'll name it chromosome 17. And we can use this function to write it into a zipped FASTA file. This is a very common format that you would use even if you download the genome from somewhere else. Most of the alignment tools will use this format. Again, this is a process that's going to take a little bit of time. We could, calculate the GC content of the chromosome 17 sequence. basically anything that we did with the biostrain object, we can do now with the actual sequence, with the actual sequence of the whole chromosome. Another quite useful function is called getSeq. So get the sequences from our genome. So now we remember we got the promoter regions, all the promoter regions on chromosome 17. And we use this function to actually get the promoter sequences from for these particular promoters. And their width are all 6,000 because we went 3,000 up and downstream, and these are the sequences of the promoters. Now you can do that, of course, with like enhancers, your protein DNA binding protein binding sites. You could use this information to look for motifs, calculate letter frequency, and everything. So in short, the BS genome stores the actual genome sequences, and you can get sequences from these packages through R. Now, finally, I'm going to quickly talk about this annotation hub resource. Now, this is actually a web service, and the library annotation hub provides you a client interface to interact with this very large online database. So to use it, you have to start by initiating a annotation hub instance. So you can just run this command here to start this instance. And it provides you with a snapshot date. So this tells you when was this version of the database that you're accessing right now is built. And this is usually in concordance with the bioconductor version and your R version by default. So if you're using an older version of R, it might be an older date. But it's very good to keep track of this information so you know that what version of data you're downloading. Now we can just take a look at what this looks like. It's got a lot of information. It has more than 50,000 records stored in this database. You can see that data provider is from all these different resources. It's got data from all these different species. It has stored data from these different R data objects. And here are the actual content of these data sets that are stored in here. And they all have an ID, which is AH certain number, and a title of explaining what the data is. You could use a single bracket to get information about a specific data set. So for example, I want to know what the number 1,000 entry in this database is. And it gives you the information about this data set. But to actually retrieve the record, you have to use this double bracket. And this gets you the actual data itself. So what's interesting is that you could actually explore this through the web browser. I'm gonna run this line here, the display command. I have it at the bottom here as well, but I want to bring it up first. So if we do this, what this does is it opens up a browser. It would open up in your R Studio window, but you could click this to open it up in the browser. And you have all the information about all the data that are stored in this whole database. And you could search it as if you're, search any web resources. If I want human data, it's just any record with human would come up. And you can search the specific columns as well. So for example, I want to search, I want only data from HG19. Oops. Sorry about that. It's not working very well for me. It should work. Let's assume that you did that. Then you would have selected the HD19 entries. So you can search all of these different columns however you want. And how do you go back to R? You could select the entries that you're interested in. I'm just randomly selecting something here. And then you can click on send. And then if you go back to your R session, you'll see that because I assigned the results back to this P object. So this were the data sets that we just selected from our web search. And then you could use these IDs to retrieve those data sets. So this is how you could interact from the web service. You could also just query it in R, like without, you don't have to open up a browser, but you can pretty much do the same thing in R. So for example, to query the database, you simply just put whatever you want to search in this query command. So this is the same as typing those keywords in the web page. So for example, I want to find all the orgdb objects, and I'm just searching anything that's orgdb. And you could actually get some information from these results as well. So if you just put a $sign, you can see, these are all the information that you could retrieve from this data. So for example, I want only human data. So what I can do is I would grab anything that has human in it from the species column, and this is what I got. And you can retrieve it like this by running this particular line. Now, in addition to these genome annotation resources, this also stores a lot of publicly available data. For example, there is a massive database or consortium called Epigenome Roadmap, where they did ChIP-seq for several histone modifications in over 100 human tissues. Now, if I want that data, I could again simply query. I'm interested in this particular histone modification mark, HCK27 methylation. I want brain data, and I want the peak files. So with all these different keywords, this is what I search for. And I can get, and take a look at what this looks like. Well, I have quite a bit of results, like a lot of files that I can download. I'm going to only get files for these two specific stages. And I want to get the AH IDs from them. So now you can see the target IDs. They're just the AH, whatever number they're the IDs of the particular data set that I want to retrieve. So now I can actually retrieve it by using these double square brackets. Because I've done this before, so this is really, really fast for me because it's just loading from cache. But I'll show you what this, if you're running it for the first time, it might take a little bit of time to retrieve the data, but this is what the data looks like. It is a broad peak format. If you've done ChIP-seq analysis, you might be familiar with this format, but it contains, sequencing names, the genome coordinates, strand, name of the particular peak. There is a score, signal intensity, p-value, and adjusted p-value. So this is the file. This will be the same as the file that you download online when you use UCSC or other databases. The advantage here is you could actually do this programmatically, and you can download multiple files at the same time, and they're all been already formatted into this easily manipulable R object that you can use for other downstream analysis. So these are the annotation resources that I wanted to cover in Bioconductor. Again, you could, I will share with you the link of these slides later. And there's some links here to, to the additional documentations or tutorials, which you can read to learn more about these different annotation packages. They are quite useful as, like, first of all, when you use some certain analysis packages, those packages may load these annotation packages for you. So just by knowing how they work would help you understand how other packages work. And if you're writing your own function, or sometimes you might be writing your own R package, it's also very important to know that you could get these annotation data easily from R, and so you don't have to download and compile them and make them compatible with R yourself. Okay, so I'm going to just jump right into the next section. So you can open up the script 03 genomic ranges R. Feel free to type in the chat if you have any questions, or you can email me later after the tutorial if you have any questions. If you didn't manage to finish running all the code, you might not be able to run through the whole scripts, but you can do so offline at another time, and I hope that the scripts should be relatively well explained, and you can always use the R help function to find the specific explanations and parameters to all these different functions. So in this next section, we're going to learn about a very fundamental and very important data type in R for NGS analysis. It's called genomic ranges. We've already seen them many times. This G ranges object, we saw it in even with our first section of the data, and then again and again everywhere. So they're basically used by quite a bit of R packages whenever it comes to manipulation of genomic ranges. And simply by name, these are an object that represents genomic ranges. So if you've managed to generate this chromosome 17 promoter object before, we could get a subset of promoters. So I just call them my promoters. And then we can use the class function to examine the class or the type of object that we have. Now they are G ranges defined by this genomic ranges packages. Sorry, did I not load? You haven't. If you didn't load the genomic ranges packages, it might have been loaded by other packages already, but you could load it just in case. And what does it look like? Well, we've already seen it many times. It seems like all of these objects have some common elements, which is the sig names, ranges, and strand. And then it's got separated by this dotted line, and it's got some extra information. So if you go back to, this is the peak that we were looking at earlier, also had the sig names, ranges, and strand, and a bunch of extra columns. So this is basically how a G ranges object is constituted. This is basic part and these additional parts. So the basic section of it is built upon this I ranges object. Now this is a very, very fundamental structure in Bioconductor, and it simply defines integer ranges. If you still have this object available from our first section of the tutorial, you can see that it is a I ranges object. And what does it look like? It's got a start and end and width, and that's it. Because I ranges defines only the ranges, so it doesn't have the chromosome names. It can be used to represent other types of data as well, not necessarily genomic data. So Some of the fundamental, I guess, principles of these type of object is that first of all, it's one-based. So coordinates always start counting at one. If you are familiar with other genomic file types, for example, bed files, you know that in bed files, the coordinates is zero-based. So there's a difference. In some type of bed files, the coordinates might be zero-based. So there might be a discrepancy, depends on where you get those specific coordinates from. Now, if you're doing analysis that requires base pair precision, you have to be aware that whether the source is one-based or zero-based, it could be all wrong if it's one off. The regions that are defined in these genomic ranges, no matter is on the positive strand or the negative strand, is always ordered by the numeric value. So it always orders from the smaller value to a larger value. And it's also a closed region. So it includes both the start and end. So if a region has like the same start and end, it'll have a width of 1. This is also different from the bed file format that I was talking about. So again, something that you want to keep an eye out if you are doing analysis that requires base pair level resolution. I'm going to introduce some of the common methods on these G ranges objects. So first of all, we can start by subsetting them. You actually subset them by using only one dimension. So although it looks like a table, it looks like it should have 2 dimensions, but to get the certain entries by index, you actually subsetting it as if it was a vector. So it's one dimension, you get the first 3 entries. When you do subset it by 2 as if it was a table, what you are getting is the metadata columns. So by doing that, you can see that we got the number, the 2nd to 5th entries, and we also obtained, we obtained only the symbol column instead of all of the metadata columns. You could name them. So right now you can see that the position where the name would be is just numbers. You can name them by transcript name, for example. And now they are like the name position is now changed to transcript names. And once you name them, you could subset them by name instead of by index. So how do you actually get to these the sequence names or the ranges? You have to use the specific functions to access these ranges elements. So you can use this function to get the sequence names. You can get the start and end position or the strand positions. Now note that for both the sequence names and the strand, they're actually stored as a factor. So it has, even though we only have 1 chromosome in our data, we only have chromosome 17, but we built our data from a resource that had all the human chromosomes. So our promoter object actually conserved all those different sequence levels. Now this could be convenient in certain cases and it might be annoying in other cases. So you could just be aware that this phenomenon exists and you can modify it if you want to. And so this is how you access the ranges element. So how do you access the metadata elements? You could use this mcalls function, and this just retrieves your metadata columns as a data frame. And then you can just manipulate it as for any other data frames. So for example, I could change my metadata and I only want the transcript name and symbol columns. So by doing that, I go look at my promoters again. Now I only have transcript name and symbol because I've reassigned my metadata. You could, of course, access it using the $, as sort of as how you would treat a data frame. So for example, we can get all the transcript names using the $sign. There are additional functions to explore G ranges. For example, you can simply get the width, the length. Now, this might be a little bit confusing because when we talk about length, sometimes we might assume that is actually the length of the sequences, but this actually gets you the number of sequences, or sorry, the number of regions that are stored in your G ranges object. Because I only got 5 entries, so it's only 5. Then there are certain very fundamental methods that you could apply on the G ranges object, and I'm going to cover the 3 types. So first of all, you have these intra-range methods. So these methods are applied to every range in a G ranges object. And you can do !, intra-range methods to see, can click on the ones that are in package genomic ranges, and you see like a bunch of them. I'm going to show you how they look like. So for example, this flank method actually gets you the coordinates that's flanking your defined coordinates. So I have my promoters, and by using flank 10 base pairs, I'm actually getting the 10 base pair upstream from my start site. Now, it's kind of hard to compare back to the original entry. Now, this is the original. You can see it starts from this 118, and it's now we're getting like 108 till 117. So that's 10 base pair upstream. We could do shift, which shifts the whole coordinates by, whatever base pair you want. You could do negative, so it goes towards the start, or you do positive, it goes towards the end. You could do resize to change the size of your regions. So for example, by doing this, we're getting, so you can specify, fix at start. So fix at start is 118, get only 100 base pairs. And you can see from the results that we only get 100 base pairs using this resize function. So using that, you can change each of the regions in your G ranges object. Now, what if you want to compare between the ranges? You have these inter-range methods. Again, you could ! inter-range methods to see a whole list of them. I'm just listing some examples here. For example, reduce would collapse the overlapping ranges. We actually have a entry like number 2nd and 3rd, they're actually the same range. And you can see now that is collapsed into one entry. There are also gaps, which finds the all the gaps between your ranges. Now we can see something quite interesting here. We only have chromosome 17, but it's getting us chromosome 1, the whole of chromosome 1, chromosome 2, every chromosome. This goes back to, if you remember, that we actually stored the information of all of those chromosomes and carried it through in our G ranges. So when we ran this command, it actually finds these additional chromosomes that even though we don't actually have the data for. So again, this is something that could be useful sometimes, but it's annoying in this particular case. Same with this coverage object, this coverage function. We're also getting all these extra different chromosomes. We could change that. Yeah, here. Okay, skip this line. We could change that by running this line here at number 50, is by just simply assigning chromosome 17 to our sequence levels. So we're forcing it to only look at chromosome 17. And now if we go back and run our gap and coverage command, we can see that we're now restricted to chromosome 17, and we're just getting the gaps in between our defined regions on the whole chromosome 17. And then for coverage is just finding, the coverage or overlaps along the sequence. So along the whole chromosome 17, we have, 250,000 base pairs that had 0 overlap, and then 6,000 base pair got 1 overlap because we got 6,000 base pair wide regions. So this could be all quite useful in certain cases. Now, finally, there is arguably the most frequently used methods is the between range method. So we're calculating the relationships between two G ranges objects, for example, like overlaps. Again, using ChIP-seq as an example, we get all these ChIP-seq peaks for one factor, and we may have a set of peaks for another factor. And often we're asked, how much do they overlap? Does this factor behave like the other factor? And now this is where we could use these functions to answer that question very easily. So the particular question that we're asking today is that, remember those peak site we got from the annotation hub? We got some peaks for H3K27 methylation. This is a marker for active enhancers. And we want to ask, how much does this active enhancer marker peak actually overlaps with promoters? So we're overlapping between this set of peaks and our chromosome 17 promoters that we got earlier. And we can do that using this function called find overlaps. The output of find overlaps is somewhat a weird format. It's called an hits object. It looks like a data frame, and you could convert it into a data frame easily. But what it does is it has 2 columns of informations: the hits from the query sequence, or the query set of regions, and the hits from the subject set of regions. So you could simply match them, that the entry number 8 from our peaks overlaps with entry number 1,100 from our promoters. So this is how you match between the 2 sets of G ranges that you are overlapping. Now, how do we actually translate this to our original peak regions, right? What we need to do is we need to get all these index and then use this index to subset our original region. And to do that, there are 2 ways. You can use a function called either from or to. So from gets the first column and to gets the second column. So we want to first find out which of our peaks overlaps a promoter. And by running this, we're getting all the indices, and we are subsetting our peak set with these indices. And what we can also do is converting this overlap object into a data frame, and then we can just get the column that's called query hits, and this should get us the exact same results. Now we know that these peaks overlap with promoters, but we want to know what are the genes that they're overlapping exactly. So we have to retrieve the information from the other data set. We have to know which promoters are overlapping with these peaks. So we just go the other way around, and we get the promoters that overlap the peaks. And then we attach the metadata from the promoters to our metadata of the peaks. Now, this is kind of hard to explain, but the code looks a little bit ugly, but the end result is that we still have our peaks, up to the Q value column. These are all our familiar format of our ChIP-seq peaks, but now it is attached with the transcript ID, transcript name, and gene symbols that we got from our promoters. So now we actually know which peak overlaps with the promoter of which gene. And if we're actually doing a real experiment, we could take these genes for a path re-enrichment analysis to see the function of our factor of interest, and so on. Okay, so there are other variations of this overlap method. The find overlap gets you sort of the most information. There are also functions such as count overlap. Whoops. So count overlap gives you this giant numeric vector, where it just tells you for each entry in your peak set, does it overlap? How many of the promoters does it overlap? So, for the first one, no promoters was overlapping. For the 8th entry, 4 promoters are overlapping, and so on. There's also subset by overlaps. Now, this is useful when you don't need to merge the 2 overlapping sets together, but you're only interested in finding out which of the regions from one set overlaps with another set. So you can do subset by overlap, and it returns only the ranges in the query set that overlaps with the subject set. There are another set of very useful methods called the nearest methods. Now, by definition, it finds you the nearest entry from the query set, from the subject set for each range in the query set. So again, we get a giant numeric vector. There are a lot of NAs because remember that the peak contains data from all different chromosomes, but our promoter is only from chromosome 17. So we're only going to get results from chromosome 17. That's why there are tons of NAs. So basically for, peak number, so this peak here would, the closest promoter to it is the entry number 1106 in our promoter list. And this is kind of hard to read. So there's another function that is called distance to nearest, where it's returning this hit subjects again. Very familiar format. Looks, we can, get the information from it was the exact same way as before, but just in addition, it got a extra metadata column, which is called distance. So this tells you the distance from the query set to the subject set. So for some entries, you can see, for example, for this one, the nearest promoter is still 200 KB away, versus for some other cases, they overlap perfectly. Now this is again also quite useful when what you're trying to find is not overlap, but you're trying to find certain sets by distance. And there are other variations to this method. Again, you could just question mark nearest methods and then read all about the details of these particular methods. Okay, so that's what we have for covering the genomic ranges objects. Now this is again a very fundamental objects that if you want to use R for more customized NGS analysis, then you should be very familiar with that object. For the final part, I put together a tiny workflow in R. I don't know how much time we have. I know there's a Q&A session later on as well. So I'll try to run through this part, maybe without getting into too much details. But basically, this workflow actually contains a lot of the steps I normally wouldn't do in R. So full disclosure, I have not actually routinely used some of these packages. However, it does provide you an option where if R is the best platform or framework that for you, then you should explore these options and know that they're actually possible by using R only. The 2 packages that are pretty useful for dealing with NGS data are called short reads and R subread. So we'll go ahead and load these 2 packages. So for the short reads, it's more useful for inspecting and quality control of the raw FASTQ files, versus subreads provides you functions with alignments and gene quantification and so on. Now, I actually included 2 very small examples, 3 small FASTQ files in the downloaded data. Now, this is the actual data from our lab. It is ChIP-seq data of RALA, which encodes the core subunit of the nuclear factor NF kappa beta. There are 2 replicates that have been performed, including one input as controls. So it's a kind of a typical experimental setup for ChIP-seq experiments. Of course, only a very small subset of the reads are used here as an example. However, it is possible to actually run this on a full library, even on your laptop. So first thing I want to do is to actually find out where are these FASTQ files. So I use this dir function to find the files that end in fastq.gz, so they're gzipped, and I get the full path to these particular files. So this the 2 replicates and the input. Now, some of the functions in this short read package are actually quite convenient to get a quick inspection of the FASTQ files. For example, we can count the number of reads that map to that are in each of these FASTQ files quite quickly. And these are the number of reads that we have and the number of nucleotides. Now, if the FASTQ file is a normal size FASTQ file, we probably wouldn't want to read it into R, as it's really usually very massive, but these files are quite tiny, so we can actually read it into R and see what it looks like. It gets read into, I'm using this function from short read, so it's got read into this short read format. You could actually use these particular functions to retrieve the information from the FASTQ file. So for example, by using this sread, so you're retrieving the actual read sequences. And again, it comes to this very familiar object, DNA string set, and these are the actual sequences. You could also get the quality of the FASTQs. Again, we know that the quality of these FASTQs are encoded in a certain way, and you can translate it into numeric values by converting it into a matrix. So now that these values all get translated into a number that is much more easily to interpret, and I'm only getting 2 entries and 40 spots. All right, so the first step, as I mentioned at the start of the tutorial, is for us to do some quality assessment. There's a function called QA for quality control. This is different from FastQC, which is literally the most commonly used tool for FASTQ quality control, but it sort of gets you similar information, and then you can browse the report using this function, browse URL. I'm going to skip the browsing part, and assuming that we've got good quality sequencing, now the next step is actually to prepare for alignment. And usually before you could actually do the alignment, you would need to generate an index for the corresponding genomes. The genome itself is very large. So for a certain program to perform the alignment algorithm efficiently, usually an index would be required so that they can access the information stored in the genome in a much faster manner. So we create a directory to store the index files. So what are we aligning to? We're aligning to that chromosome 17 sequence that we got earlier from the BS genome object. So we're using this function called build index. And this is the index name that we're building. This is our reference sequence. We're using this gapped index, which practically is just faster to build, but you can read upon it that essentially it's just building an index, leaving certain gaps, but making the process a little bit more efficient. Now, this should finish in relatively short time. If you're using, if you're not using R, you're just using any tool for alignment, every tool would have its own index building process. Also, with different versions of tools, you should probably build a different index with the different versions. So this is something that you should be aware of and before you do any alignments. Right, so this was successful. Now that we have the genome index built, we can actually perform the alignment. And again, we create a folder for alignment. This is also a little bit of a trick or something that you would want to do when you are doing your analysis. The real analysis is having different directories for different analysis. You can rank them by number or name them with a number so that it's easily, it's clear that which step you did first and which step follows. It's better than having everything in one directory, which might get super massive after you complete the whole process. So we're using this function called align, and we're giving it the index, the location to our FASTQ files. We're telling it that this is a DNA sequence. We're telling it what the file names of our output should be like, and some additional parameters. Again, these are the very common parameters that you actually would give any alignment tools or any alignment softwares. Now this is in R, we're running the R function, but on the command line, these will be different parameters that you pass to the tool that you're using. On Galaxy, these will be different parameters that you can select. But fundamentally, you have to give it the genome that you're aligning to, the FASTQ files, and the output names. Those are the most essential parameters for every tool possible. So this package is actually able to perform alignment quite efficiently. If you've built a gapped alignment, if you've built a gapped index, it actually slows down the alignment a little bit later. So there's a trade-off. You could spend more time building the full index, which will make your alignment faster later on. While we wait for the alignment, again, like these options, these options are available in R, but they may not be the best performance for your particular type of data. So you should always research what is the best practice for the particular type of data that you're dealing with. What's nice about it is produce a very clear and simple alignment log that tells you the total number of reads, number of mapped reads, uniquely mappable reads, multiple mapping reads, and so on and so forth. So after the alignment, what we get are the type of format called BAM files, and this is the file format that we use to store sequence alignments. In R, there are two packages to deal with these BAM files. One is called RSAM-tools. We could use it to sort and index our BAM files. On the command line, the most commonly used tool is called SAM-tools without the R. And this is sort of an R version or R equivalent of SAM-tools to perform some of the similar analysis. The other package that is very useful is called genomic alignments. Now this function, from using this package, we can actually read BAM files into R. For example, I'm just reading in one of the BAM files from our alignment, and you can see that this is the sequence chromosomes start and end, or width of the reads. So CVAR's sequence tells you the matches and mismatches and everything. You could use the flags. There are ways to filter the reads that you read in. Again, I'm going to skip that, and you could look at the documentation for more specific examples. Now that we have our reads aligned, the next step for ChIP-seq analysis is usually peak calling. So to find where in the genome do the reads cluster comparing to the controls. So for this task, the most commonly used package or tool is called MAX. And here I'm using an R package. That essentially does the same thing. What it does is it bins the genome or bins the region that you're interested in into certain bin size and put it into 200 base pair bins. And then on each bin, it counts the number of reads that maps in your experiments and their controls, and then it fits a statistical model to find the bins where you have an enrichment over the controls, and then it merges the overlapping bins, refines this process, and produces your result. Now, I have this function here which makes it a GRanges object. We can just quickly see what it looks like. This is what the GRange object looks like. And these are the coordinates of the peaks that we actually found. And you can export it into a BET file format for subsequent uses. And then finally, I want to just touch upon visualization in R. So in the previous session, you learned about how to visualize your data on the genome browser. There are clear advantages of using a genome browser. You could screen through the whole genome. There are a ton of publicly available data on the genome browser that you can compare your data to. However, when it comes to making figures, sometimes genome browser could be difficult. Let's say you want to visualize 100 spots in the genome, where your PI wants you to show him 100 spots in the genome. With the genome browser, you have to manually put in the coordinates and take a screenshot. So this is where being able to do it programmatically in R could be advantage, or at least being able to have that option. It also helps you with formatting the plots the way you want. You could change colors, font size, how you want to organize it, and everything. So one pretty good package in R to do that is called gviz. I'm going to just blindly run through all of this code and show you what the result looks like, and you could sort of study the code yourself later on. Essentially what it does is building each different type of tracks, and then ultimately it can use this plot track function, attaching all these tracks together to make it one plot. And it takes care of the genomic coordinates. So if you were to make the plots yourself, you might have to worry about lining them up perfectly, but with this package, it takes care of all these details for you in a coordinated fashion. So here I am showing you, this is the genomic coordinates. The blue bars here are the peaks that we called. This is the gene model that is annotated here. The orange track here is the actual signal of our ChIP-seq experiment versus the gray track here is the signal from input. So you can see that our peak calling really works, and this is indeed a pretty good enrichment compared to the controls. So essentially with this package and with all the things that we've learned today, you should be able to produce plots like this in customized regions, and you should be able to change the appearance of these plots with a little bit more study. So that concludes our tutorial, and I just want to take a tiny little bit of time in the end to say that when one challenge with NGS analysis is that you actually have way too many options. There are new tools, new pipelines coming out almost every day. So one suggestion is that you should always read the vignettes or documentations very carefully. Make sure that you know what you're doing instead of just simply following certain tutorials. Like instead of following them, just following them, you should really know what each step is actually doing. If you are new to a certain type of analysis, try to find tutorials or pipelines that are most widely used. They're usually most widely used for very good reasons. If you were to explore different tools, you'll realize a lot of the bioinformatic tools aren't very well developed. Might be very difficult to use or have poor documentation, and those things that and we don't want those things in our pipeline. So choosing the most commonly used ones is usually helps you to avoid that kind of problems. I've also listed a couple of useful tutorials, and these are also good starting points when you're completely new to the field and just want to start somewhere. Right, so with that, thanks everyone for attending today's tutorial. It is very long, so thanks for sticking till the end. I believe the link to the slides and will be sent out later, and so it's got my email on it, so feel free to email me if you have any questions after the tutorial.
Thank you so much, Dr. Ho, for the presentation. It was very interesting. So if anyone has any questions, please feel free to write them in the chat box, or you can just unmute yourself and ask your questions. But other than that, if anyone has any questions, please say so. But thank you, Dr. Ho, for joining us today, and I hope everyone learned something that you can take home with you. And also, before you leave, please fill out the feedback form that I put in the chat. I'll paste it again. Just a second. Yep. And as Dr. Ho mentioned, we will be uploading the slides and the recording, and we'll send you the link to this material through Eventbrite. So yeah, if you have any other further questions, please feel free to ask. If not, we hope you have a great semester. Thank you so much. And thank you again, Dr. Ho, for this presentation.
All right. Thank you.